Have you ever thought about how the sugars in your food is analyzed and measured? This can be done using high-performance liquid chromatography, which is an analytical instrument that used in separate different types of sugars such as sucrose, glucose, fructose in the food sample, like milk, chocolate, juices, and cereals. Any HPLC will consist of solvent reservoir, which is a mobile phase, pump to produce a higher pressure, sample injection, column, which is a stationary phase, detector, and computer system. So, how HPLC works? First, prepare your sample according to a certain standard method that is used in the analytical science related to foods. Here, AOAC International Standard has been used to prepare the sugar sample. Then the sample filled in a small vial, and now it's ready for the analysis. Second, select your mobile phase. The mobile phase in HPLC is a mixture of solvents where the analyte should be dissolved in. Therefore, make sure that you select the appropriate solvent. Mobile phase is used to push the analyte through the column. However, since our wanted analyte sample are sugar, in particular polar sucrose, the solvent must be polar to allow the sample to dissolve in it. So here we use acetonitrile and water as a mobile phase to carry the sample. Third, select the column. The column is another important component you have to choose before the separation process starts, because it contains the packing material needed to perform the separation. The particle size in HPLC column is small, so the analyte needs more pressure, which will be provided by the pressurized solvent to go through the column and separate them. The columns are made from cylinder of stainless steel and contain non-polar hydrophobic amino particle. But why non-polar stationary phase? Let's talk a little bit about it. In HPLC, we have two phases, normal and reverse phase. Here, the reverse phase is used for sugar analysis. In the reverse phase, the mobile phase is polar solvent passes through the stationary phase that contain non-polar material. Each type of sugar has its own polarity and reacts differently with the amino particle. For example, fructose is more polar, so it will not bind with the non-polar molecule of amino, and it will pass quickly through the stationary phase, and it will elute first. Now, how the separation process occurs? The sample is injected automatically by using auto-sampler into the instrument, specifically in the beginning of the column. Then the solvents are flowing continually in the device using pump to be pressurized. In the sugar separation, the isocratic pump is used where it consists only one solvent. The pressurized solvent carries the analyte through the column. Inside the column, certain interaction occur between the mobile phase and the stationary phase. The strongest binding molecules are eluted from the column at the last, while the weakest will elute the first. For each analyte, there is a specific time indicate when the analyte has been eluted from the column, called retention time. Retention time can be used to determine the identity of the analyte being used. And now the analytes are ready to be detected by using certain type of detectors. The detector that used to detect sugar is Refractive Index Detector. This type of detector can measure the ability of the molecule to refract the light beam or change the light's angles as it passes through the different materials. Then the refracted light beam signals is converted to electrical signal, which the computer can understand. These analytes can be illustrated in the chromatogram, which is a plot of retention time and the intensity of each analyte. For each analyte, there is a specific peak that can be used to determine the identity of these analytes, and the concentration can be measured by calculating the area under the peak. And by these steps, the sugar can be separated and analyzed. Thank you for watching this video. Hope you find it informative.